The cardiovascular system sustains life by delivering oxygen and metabolic substrates to all the cells in the body, by removing end products of metabolism, and by carrying our internal defense system, the leukocytes, always on the lookout for pathogens to be destroyed. The cardiovascular system also carries platelets and clotting factors that are ready to react to any type of injury. The endothelium has developed specialized homeostatic processes that regulate the movement of water and metabolic substrates into and out of the vessel. In the lungs, alterations of endothelial permeability result in increased amounts of fluid accumulating in the interstitial space. This fluid impedes the normal exchange of respiratory gases, and in severe cases results in respiratory failure. The normally antithrombotic endothelial surface is covered by the glycocalyx, a filamentous layer 1 micron to 500 nanometers in height. The glycocalyx is composed of proteoglycans, and primarily their long disaccharide chains, known as glycosaminoglycans, that form the protective layer on the cell's surface. Functional components of the glycocalyx include heparan sulfate proteoglycans, chondroitin sulfate proteoglycans, and hyaluronin. The glycocalyx is a negatively charged layer that binds serum proteins. It is believed serum proteins fill the interfiber space, creating a gel-like layer on the endothelial surface. The glycocalyx and absorbed serum proteins have been termed the endothelial surface layer, or ESL. This ESL provides a formidable barrier to the penetration of water and serum proteins escaping from the vascular lumen, and therefore plays an important role in permeability. A specific functional attribute of the glycocalyx is its participation in mechanotransduction. These filamentous projections can sense changes in blood flow and pressure, regulating signaling in endothelial cells and underlying smooth muscle cells to control vascular tone. One of these signaling molecules is a gas called nitric oxide, which is produced by endothelial cells in response to a variety of stimuli. Increases in flow and pressure stimulate nitric oxide production through a heparan sulfate proteoglycan-dependent mechanism, and the nitric oxide contributes to increased permeability. Inside the endothelial cell, nitric oxide is a reactive compound and rapidly reacts with cellular proteins and can alter their function. Nitric oxide nitrosylates many proteins, including VE-cadherin, a protein important to creating the cell junction. Nitrosylation of VE-cadherin is a signal for cadherin to be removed from the junction and targeted for degradation. The loss of VE-cadherin is associated with increased permeability and allows fluid to leak through the cell junction. Pressure-dependent changes in endothelial permeability add a new level of complexity to our understanding of fluid balance across the vessel wall. The realization that pressure can alter endothelial permeability through a process of mechanotransduction creates new paradigms to understand the etiology of pulmonary edema during acute heart failure, fluid overload, and other states where pulmonary vascular pressures are acutely elevated. During acute inflammation, cytokines and other inflammatory mediators cause the glycocalyx to be shed. It appears that cytokines activate proteases that cleave the core protein and result in fragments of the proteoglycans being shed into the blood. The breakdown of the glycocalyx has important physiological implications. Adhesion proteins that support the binding of white blood cells and platelets are exposed, allowing these two cells to bind to the endothelium. This binding triggers the release of additional inflammatory and procoagulant mediators that propagate the inflammatory reaction. Developing a greater understanding about the glycocalyx and how it regulates cardiovascular physiology may identify new targets for therapeutic interventions.